Okay. So since we only have an hour here left for a little bit of a roundtable discussion, we're gonna just open it up to some questions. So either send in your questions through the chat, or since we have a smaller group, I'm gonna do something that's a little unprecedented. I'm just gonna unmute everybody, so you could just have like a little bit of a discussion. And if it gets out of control, then we'll adjust from there. And so we can have a little bit of a discussion that way too. So let's see here. So Steve Riley just asked the question, are there any comments regarding VSH being genetic or aspects learned? So is it a genetic component or is it a learned behavior? Garrett, you may want to touch that one a little bit. Everybody could unmute themselves. You should be able to unmute yourselves. Let me just see here real quick. Okay. Let me just see here. Let me just do something real quick. Try it. Actually, try to unmute right now. I think I just reversed.
Okay, that worked.
There we go. Sorry for my technical naivete. But yeah, everybody should be able to unmute yourself. So if you want to ask a question, please just use that or type it into the chat box. But yeah, so Garrett, we had that question about VSH. Is it genetic or is it learned? You want to speak on that?
Yes, it is genetic. We breed for it. So we've determined through the different experiments we've done that, yeah, it is genetic. You can see when it gets outcrossed, the trait diminishes fairly quickly. But if you select for it, you can select for it so intensely that they will almost cannibalize all their brood. So it is a genetic component.
Okay. Yeah, so Steve asked another good question. So looking at what proportion of workers need VSH qualities, can we do open mating to make VSH effective?
I think that you need around 75% of the workers need to have VSH genes in there to be able to keep the resistance to a level that will enable you so that you don't have to treat the bees with chemicals. So, and that could vary between climates, like in the north, maybe that doesn't have to be as high because they quit brood rearing during the winter, where in Hawaii, where they raise brood all year long, it has to maintain that 75% or higher.
Yeah, and so I think open mating can be a viable strategy because the one project that we started about, what was that, in 2017, we used Paul line bees. Garrett showed some of the data from that, looking at the mite levels and the honey production. Those were Paul line queens that were produced via open mating. So here at the lab, we have a large number of apiaries that are drone in inundation areas for Paul line. And when we use those in that experiment, they performed quite well. And what was really nice about that is that, yes, we can produce viable, highly effective Paul line queens via open mating. So it's not just a product of an intense instrumental insemination breeding program. This can actually be done at the commercial scale. So hopefully that's what's going to be going on out in Hawaii with the Hilo bees. Yeah. Another thing about VSH, VSH is a very complicated behavior. There's a number of stereotypical steps involved, which involves detection of an infested cell, opening it up, sensing that varroa from the brood, removing the brood, or recapping it if there is no varroa in there. So that complicated behavior begets the fact that the genetics of it are a little complex. It's not just one gene, one behavior, because there's so many different pathways involved. So the pathway for VSH behavior is quite complex at the genetic level. And that's why Garrett was saying that how it goes away after it's outcrossed for only a few generations. So that complex trait needs to be reinforced every time just to maintain that each gene that's involved with this behavior is maintained.
Frank, if I can talk a little bit about that too, and maybe answer
Please, go
some
ahead.
other questions about the natural adapted stocks. So one of the things there is that, like, it's not only the trait itself, it's actually the genetics of honeybees also that contribute to this sort of rapid decay of particular traits. And so honeybees tend to be relatively unique because they have a very high recombination rate. So that means that effectively they can disassociate portions of their genome like quite fast. So that means that like every SNP tends to add more or less independently. To give you a perspective, that doesn't happen much with other organisms like cows or even us, for example. We're comparatively speaking a little bit inbred in terms of that. So when you breed honeybees, that there's benefits and then there's cost to this. So the benefit is that you can actually breed for traits in honeybees quite faster than you would for any other organism, and we've seen that happen time and time again with high and low pollen lines, for example. That's a Rob Page bred for those traits. For only 5 years, he was able to get quite a high amount of distinction between those two lines, and then he had to repeat it again because of politics in terms of moving from one university to another, and he was able to successfully achieve it again in 5 years time. So you know, and then there's examples from China where they are breeding for high royal jelly producing colonies, and they were able to get a 10-fold increase in a matter of something like 10 years or something like that. The cost to that is that the moment you start outcrossing, that trait sort of becomes modeled relatively quickly, unless it's a particularly critical trait, right? Or you know, and as long as it's not in a place where there's a lot of other bees that don't have the trait, right? Which is usually what happens here. And so that's the type of concern that we have with open mating, but and how the genetics kind of maybe contribute a little bit to that difficulty. With regards to naturally mated stock, it's worth pointing out that VSH bees and pole line and all these lines that we're talking about, they were natural stocks, like VSH started when John Harbaugh went and sort of ascertained some of the colonies we keep in the lab and just looked for ones that were resistant. So they were part of the broader population of bees, right? So when, you know, what can we learn about them? Well, we've learned a lot from them because we developed these research lines, right? And yes, there are other lines that are VSH, that have VSH and that are resistant. But those lines are a little bit further along in terms of being examined when compared to pole line and VSH. So we're kind of like, you know, we only have so much attention and so much time and so much money. so that's why we focus on certain things.
So Steve asked another question. What's the trigger for uncapping? Is it chemical or movement? Actually, it's been pretty well shown that it's a chemical cue. It's a chemical called Z10-triacontine. Probably never hear that word ever again. But it's this very specific chemical that has a very specific chemical makeup that they respond to. If you make the chain bigger, smaller, they don't respond. It's this very specific compound. Kara Wagner in Olav Ruppel's lab has shown this, and they published the results this year. It is pretty convincing, but it's still only part of the story. There's probably, as I mentioned, the behavior is pretty complex, so there's likely more chemicals that are involved with this behavior. But that seems to be a major one in this system, this Z10-triacontine. So, and yet that's the important thing we have to understand, is that we, in our work, is looking at the effects of these chemical signals in our research. So whether it's looking at things like VSH or in some of the other research that we've done, we've seen that just the different chemical environments within a colony can have an effect. So Mike Simone-Finstrom and I have looked at the effects of propolis on honeybees, and we've been finding that they can alter their sensitivity to pesticides just a little bit, not very much, but it's something that we're working on and hopefully going to be publishing very soon. So there's all kinds of other things going on with what's going on in the hive. So a lot of this chemical cues do have tremendous impacts on the way honeybees act. So, and we try to do these evaluations at the field level, which is kind of a difficult issue to deal with, because if we're only doing this research locally, we might get results that are only supported in Louisiana, let's say. But we are very fortunate here at the lab that we have great relationships with commercial beekeepers in Louisiana and adjoining states, but also up in the Dakotas, out in California. So, and even as Garrett was saying about out in Hawaii. So we really leverage those relationships to do actual field work in the field as that are going to have real world impact. So we have to validate and reinforce those findings. So we do travel a lot for our work to do these evaluations in the field. And those projects, they tend to last at least 1 or 2 years. So you can imagine the duration of how expensive it is, then also just the traveling and the sampling and everything like that. So these things take time. They are very difficult to do. But it's just one of the goals of our lab is to do that kind of research. So whether it's looking at VSH behavior in real world situations or pollination services, these are a lot of things that we have to keep working on. Okay, so Danielle asked a question: is there any genetic evidence or understanding of locally adapted traits in bees?
I can take that if you want.
Yeah, go ahead.
So yeah, there's actually, when we look at broad sort of genetic diversity of bees, like bees tend to sort of segregate rather quickly genetically, depending on where they're at and the situation they're in. So what I would say about locally adapted traits, that's kind of like a loaded term in that, it's dependent on the environment that they're adapting to, right? But for example, we know that honeybees as a whole have like ancestral ecotypes, right? And so those ecotypes vary very subtly, but it's things that are relevant. For example, if you look at Caucasica, which is one of the ecotypes, one of the races, subspecies, whatever you want to call them, but they are more temperate and actually mountain sort of dwelling bees. And so they actually don't do a 1 year generational cycle. They can wait up to 2-3 years before they produce a swarm, actually, and that's because the environment that they're at in their native environment is very harsh and unforgiving. So they require that time to build up resources, and these are characteristics that are like endemic to that ecotype. So that when you take it and you move it somewhere else, they behave in the same way. So there's a genetic component on that regard. Even though they may be in an environment that actually has more resources, they still take 1 to 2 years to kind of form a swarm, right? That's at a broad level. At a specific level, we see that sort of differentiation happening as quickly as something like 12 or 15 years. So for example, there's work that I've been doing myself and with collaborators in Puerto Rico, showing that there's a specific Africanized population there that not only has a unique characteristics, but then on top of that, it seems like it's continuously adapting to that island environment. And so there's a lot of traits that go with that. A lot of them actually are things like being able to store food in case of hurricanes. So they have a more storage-oriented profile than most other Africanized bees do. And so these are traits that we're talking about, but the genetics behind them are so complex that we can get to them, but they will take some time. And many of them are behavioral or developmental, which it's another layer of information that you have to kind of sieve through. I don't know if that gets at the locally adapted concerns, but
Anybody else want to weigh in on locally adapted traits? Okay. So Steve asked another follow-up question: is the chemical trigger from the distressed larva pupa from the mating cycle of the foundress commencing? So in Kara's paper, it talks about the way that these hydrocarbons are produced. So when these larvae or pupae are parasitized or by varroa and infected with deformed wing virus does a lot of interesting things. It gives off a larger proportion. It changes the chemical signature. And so those brood are giving off a altered signal that is detected by the by the nurse bees that are working to remove any potentially infected brood. So there's a little bit of it's not the whole issue. There's a lot more that goes into it because there are other ones that so if the brood is giving off a signal, it has to be received by that nurse. So maybe the nurse has these other adaptations. So there's been some back and forth on, is it a brood signal or is it the worker signal? Both sides show evidence. I think it's a combination of both, not actually mutually exclusive. So these signals are given off when they're parasitized by Varroa and or infected with deformed wing virus. So that stimulates this different chemical cues that they use. These aren't anything unusual. These are actually a normal part of their life cycle. They aren't specifically and only due to Varroa parasitization and deformed wing virus infection, but it's one of those triggers that stimulates it. Well, that one specifically, I don't know of anything else specifically that does it, because this is kind of a nascent field, as you can imagine. Working with and identifying these chemicals that are coming off brood, it's tedious and expensive. So I don't know exactly what that is all about. Yeah. So Ariane, you mentioned something earlier about honeybee genetics being difficult. What is so unusual about honeybee genomes and their genetics that make them difficult to study?
So I mean, we know that there's like 3 major characteristics that honeybees have. I mentioned recombination rate. So that's, I mean, that seems like a daunting term, but all it is like when 2 organisms have sex, they take their genetics and they kind of shake it a little bit, so that, you know, the resulting progeny, so the resulting daughter, for example, or queen from a queen drone mating, it's actually more of a mix, like a milkshake mix, rather than just mom and dad by themselves in the organism, right? And so most organisms, they do have recombination, but it happens, you know, a couple times, let's say, across the genome when sex is occurring, right? And that's enough to generate like all the diversity we see, for example, in mammals. It's enough for that. For honeybees, they actually do that like quite a lot more times. In fact, the recombination rate is the highest of any sort of multicellular organism recorded so far. So that means that like the blender they're using is set at high when compared to other organisms. So I talked a little bit about the benefits and sort of detriments from that. The other thing that they have that makes them unique is that they forcibly outcross themselves. So they have to do this because they have a sex determination system where if you have the same type of gene in like 2 copies that are exactly the same, then what comes out of that mating is a diploid drone, which drones are generally useless. A diploid drone is totally useless, right? So like the one job it has, which is to breed and produce daughters, it can't even do, because a diploid drone, when it breeds with a queen, nothing comes out of that mating effectively. So they avoid that. They, queens mate multiple times, so they mate with like up to 20 drones, make sure that and guarantee that at least one of those, I mean usually more than one, hopefully, don't have her exact same sort of gene essentially. But it also pushes it so that the individual queen is always looking for new genetic material. And you can imagine how, if you're trying to maintain a particular characteristic and maintain your population relatively uniform for that particular characteristic, having a mating system that then is forcing, that where the queen is just looking for as much difference as possible, disrupts that. so the challenge is bees is maintaining, when breeding bees, is maintaining everything relatively the same, at least for the trait of interest. And when your mating system works as antithetical to that, then you're, that's where the barrier is. Beyond that, they have really small genomes, which means that, the blender set on high is even more impactful because it has smaller, chunks of genetic material to work in. Comparatively speaking to vertebrates or, plants or something like that, which have very large genomes and low recombination rates. Well, that more or less assures that even with a little bit of recombination, everything stays more or less the same. But with honeybees, like there's quite a lot of, it's actually, to me, every time I do honeybee genetics, it's surprising that you can get a bee out of that pairing consistently because of all these changes that can occur during just sexual reproduction. So.
Good. All right, Katie Lee asked the question. I think everybody here at the lab has I would encourage everybody here to weigh in on this question. Is what is your definition of success in a breeding program? Is it the elimination of miticide use, or would you be happy if commercial beekeepers could eliminate one treatment? What do you think is possible? So I'll start off with that. I think the definition of success is to get this material out and used in real world situations. So to translate that from our lab research to actual field use, I think that's really successful. For the VSH pollen line sort of breeding program, I think that ultimately it's going to come down to using less miticides, maybe one treatment at least, because we have some evidence that shows that on its own, it's pretty good, but a miticide treatment makes it really good. So in terms of reducing Varroa levels, we've demonstrated that it can really knock it down. And then also to pair it with enhanced survivorship. So just doing one less treatments, let's say you're a commercial beekeeper and you have 10,000 colonies. If you could reduce one treatment, there's $2. Let's use just the cost, a rough cost. Let's say $2 a hive per treatment. That's $20,000 you just saved by not having to do a treatment using this integrated VSH behavior. I think it's not going to eliminate all miticides. I think miticides are here to stay, and I think we should use them in a judicious way that, one, reduces mite levels to levels that don't lead to enhanced colony failure, but not so much where we breed for resistance and we lose the utility of it, because miticides aren't being developed much more than what's out there right now. So I think it's just a matter of just keeping things in balance instead of relying on one thing more than another, whether it's miticides or only VSH. So I think it's those things working in concert, which would be good. So we do think that is possible. Does anybody else want to weigh in on that question?
Yeah, I do. So one, what's a successful breeding program? To me, that one determines what your purpose of the breeding program is on what makes it successful. If we're looking at mite resistance, then the elimination or the reduction of using chemicals would make it successful, as long as you're keeping the traits that commercial beekeepers want, like the honey production, the large colonies for pollination. I do think it is possible to almost eliminate completely the use of chemicals, especially the way commercial beekeepers do operate, where they take colonies to the almonds, they come back and they split everything and kind of basically requeen the colonies. So you're getting that brood cycle break in to help reduce the Varroa levels. And I think we've shown that with the studies online and the initial field trials with the Hilo bees, because those experiments, those colonies went through an entire yearly cycle with no treatment at all, and they survived and still maintained low mites.
Anybody else want to weigh in on a successful breeding program?
Sure. I can just say that, I agree with both Frank and Garrett that, ideally a successful breeding for Varroa resistance would be that you don't need to use miticides as a regular thing, but maybe as a spot treatment for, an infestation here and there. And, maybe that's, kind of a pie in the sky dream for a widespread thing like that. But if we can get to that point, I think that would really be a successful program.
Something else with the breeding too is, to me, because the breeding that I do, I try to gear towards what beekeepers want, what commercial beekeepers are looking for. The other thing is uniformity in the stock. If you can reduce your labor costs tremendously as a commercial beekeeper, if you can treat everybody basically the same at the same time each year, or when you're going out into the field, you can, honey production is about the same per colony. You can super all the colonies instead of saying, well, this one needs one super, that one needs two, or let's do mite washes, and well, I have to treat this colony and that colony. Well, commercial beekeepers don't do that. That's why they just treat everything. And so if you can make it more uniform so that they can reduce on their labor costs, then that also will make the breeding population successful.
Yeah, that's something that needs to be thought into those calculations. Good. Okay. So Danielle had a few questions here. Do you have any metrics for translation? How many beekeepers, colonies, queens, etc., for Russian Pauline? So I think a pretty good translation that we've had here at the lab was the creation of the Russian Bee Breeders Association. So that's kind of an autonomous group that maintains Russian stocks and is validated here at the lab using genetic markers. How many people are there in the Russian Breeders Association? Lilia here. Lilia, you might want to, could you weigh in on that a little bit?
Oh, there right now there are at least 16 or 17 members. I guess there are 3 additional new members.
Okay.
Yeah.
And about how many queens do the Russian beekeepers raise annually?
Oh, yeah, that's a good question. I know they're limited to their, I guess, when it comes to queen production. I know Stephen Coy is going up for more than 1,000 per year, but the others are not really into queen production because they have to produce honey as well, and also packages or nukes. So yeah, I guess Stephen Coy is the biggest queen producer among the group.
Okay, good. Yeah, so for translation, I think it's really a matter of working with a lot of the other commercial beekeepers and getting them to act and to measure these colony, these stock performances in real beekeeping situations. As far as beekeepers, colonies, queens, I think when we're doing these evaluations, we really are fortunate in that the beekeepers we work with allow us to do some absurdly large studies. So the one study we did with a local beekeeper, we used, oh boy, finally in the final experiment, every year we had almost 400 colonies in the study, which is a large way of evaluating that. That's in a statistically rigorous manner, but also to a field relevant manner, because we have to deal with other variables such as apiary location and migratory pathways and things like that. So translation is a little tricky, and I think that's actually a good challenge, is to actually define what that is. So Danielle, that's a very good thought-provoking question. So that way we could have something like a yardstick to measure by, but we'll have to keep thinking about that. Then she also asked to Ariane about the bees you show look to be distinct gene pools based on your published analyses. How far are we connecting the genes to the behaviors? And is there a lot of logic tossed around there that are necessary trade-offs? What has to happen to answer with that genetic tools? So if you could read that in the chat, you could go from there. But let's see what you could think about to answer Danielle's questions.
So I'm gonna say that we actually have made inroads into sort of tying genetics to behavior. There's actually, so I mean, it hasn't necessarily happened for BSA trait to some degree. There are markers, for example, that we know correlate with the trait. The complications we had is that they were correlating with the trait in one particular population, and then the pattern sort of shifted in another population. So, that's an answer that we got. It's like, yes, we can tie genetics to the trait, but then we got another question, which was, well, is it the same for every population? That's literally what linearly led to us sequencing all these sort of stocks, is because we now can say, okay, well, these are markers of interest. Do they look comparable across these, or do they look different, and do they look comparable across bees that exhibit something like VSH or something along those lines? So that's within the manuscript is assessment of that. We didn't do in that manuscript, which we should have actually, but what we didn't do in that manuscript was actually tying in that variation from the previous publication to the broad genetic diversity. But that's also a matter of resolution. Like we were limited by some of the approach that we did. So we're hoping to get more to that. So for VSH in particular, I would say, I mean, and we're currently working with Missouri with Chris Elsik, and so we likely will have something within the next couple of years in terms of genetic markers for the VSH behavior. That's my hope. Like whether that's reality is a separate thing, because biology is biology and science is science. And so that's for that particular trait. For other traits, it's happened, right? So like I know Leonard Foster, and I'm sure Daniel is aware of this as well, was able to do that. He was able to do that a little bit easier because he didn't look at the genetic information, he looked at the protein level information. And so it's easier to build a collaboration there because the protein is really what's the final effector when you're talking about genetics, right? And so the complication with that one is that, well, just measuring the protein alone, right, or the types of proteins alone, it's a complicated mess. And you still don't know whether those markers that he found in that particular population translate to other populations, because that wasn't part of his study. So that's another follow-up that needs to happen before we can sort of seriously consider some of those characteristics, right? So that those are a bit of the complications and a bit of the framing, but the answer is soon. And I know that's a standard answer, but we're really positive that we can probably get to something within the next couple of years or so, at least for that trait. And then the question becomes, how can we incorporate it into like a breeding program, right? Which is, I guess, getting at a little bit on your trade-offs, is, you know, once we have those markers, then how do they get incorporated into a breeding operation, right? Because that would be the markers for the trait. It is not the markers for, you know, all the characteristics that you consider to be good characteristics for something like Hilo bees, for example. There's many other characteristics that are desirable about Hilo bees than just VSH, right? And so you don't want to just take the markers for VSH and just, you know, use them like I was describing, where it's just, you know, you just picking bees based on those markers, because you might be losing a lot of the other characteristics, because we're, you know, that's not a panel-based selection, that's a trait-based selection, right? And so what those panels are useful for, though, is looking for other populations that have those markers, and then kind of, you know, begin to build like a resistant population that then you can use to, similar to what we've done in collaboration for Hilo, you know, incorporate that trait into a broader sort of, you know, better bee effectively. So, and so that's kind of where the trade-offs come in, essentially. Is like, how do you plug this into a system, and then how do you plug it into a system that is, you know, a rough translation from the Spanish saying in my country, but it's like pretty cheap and good. So like those are the three criteria. So, and for bee breeding, that's the three criteria, right? You want a bee that's nice, you want it to be cheap, and you want it to be a good process, right? So, but yeah, I hope that answers some of the question. If not, we can talk more.
And even some of the performance trade-offs that we see is sometimes with pollen line, and Garrett mentioned this a little bit, they do make less honey in some instances, especially when resources are very low. They seem to produce less honey, and because you've noticed on Garrett's graph, they showed, you know, only 30 to 40 pounds of honey produced, even in healthy colonies. So the trade-off is that they don't produce as much honey, but yet twice as many survive to pollination. So if your operation is really focused in a little bit on the pollination side of it, that might be more attractive than the honey production aspect, because of the economics of having more colonies at almond pollination, let's say, might outweigh the lower honey production. So that is another kind of trade-off that we could consider.
I mean, that plugs into with any breeding decision, right? With any trait that you're interested in, there's going to be something that you're enhancing and something that, I mean, if you're lucky, the impact is minimal. But sometimes when you're enhancing something genetically, you can't help but affect something negatively, right? I'm not saying that high VSH leads to low honey production. I mean, we've shown that it's actually improved even with high VSH in the population, right? Because the Hilos have steadily improved in terms of their productivity, right? And the same thing, VSH bees, the original VSH bees were like, you know, piss poor effectively at producing honey, but the moment we outcrossed them and sort of generated pollen, we showed that pollen was a little bit better, right? So that, you know, it's not a one-to-one trade-off, But there's definite things that you're going to have to take into account, because the energy of the nurse, let's say, is investing and going around looking for sort of infected cells. His energy is not putting into making more foragers that would then go and make more honey. You know what I mean? So it's like that, those are the types of things that you are not going to be able to get away from. And then it becomes more of like, well, let's improve it as much as we can and then see if the cost benefit is still good, right? And so that's kind of where you would have to consider that.
All right, Joe asked the question, in a commercial operation, maintaining a stock or trait like pollen line is difficult. What is the lab's long-term plan to make this easier for commercial beekeepers? So this comes down to the genetics in each stock. So the more homogeneous it is, and the more, I guess the word is consistent it is, the easier it is to maintain. So it's a matter of keeping those genetic. Whoever has a dog barking, could you please turn off the microphone? So that's one important thing, is to try to control those outbreedings. Garrett, you mind taking a little bit of stab at that too?
Yes, that's kind of what we look at with the pollen also is we don't put, I mean, we put effort into the breeding, but the amount of laboratory type measurements that we do are very minimal with the pollen. So like for BSH, the original experiments with John Harbaugh was a large package of bees where they had the same mite levels split up. You know, I think they did like 75 pounds of bees split up into 30 or so nukes with queens, and then measured 10 weeks later, measured the varroa levels in the brood, the bees, all that. And they got to the point where, okay, we have the trait to where for breeding, you went to the colony and you looked at the brood for non-reproductive or for mite removal, depending on who was doing it at that time. Jeff Harris did mite removal checks, and we do non-reproductive. So that's all labor intensive. For the pollen, what we do is we end up breeding just as we would a normal queen producer would. We produce the colonies, we let them build up to full-size colonies. We take notes when we go out to the colonies. So there's notes of, oh, this one's a little testy, or I got stung in this one, or the brood looks really great in this one. Just, you know, just notes of importance. And then what we end up doing is when it comes time for the almonds, we go through the colonies and we look at the population size, because that's important for the beekeepers. And then we just do like a mite wash to see where the mite levels are. And then we narrow down. What we're doing is you're narrowing down what you're looking for. So the easy traits that you're looking for that are quick and dirty, for lack of a better word, is, you know, you go in and just, oh, this one's too mean, I'm not going to do this one anymore. This one's too small, I'm not going to use that one. And you eliminate those to where by the end, when you're looking at the mite levels, you're only doing mite washes on a certain number of colonies. And then you pick out the ones with lower mites. And then we look at the brood just to verify our findings of the low mites to make sure there is no mites in the brood, or very little, low non-reproductive. So we're, you know, part of our long-term goal is we are doing these techniques to try to also minimize the labor, and to kind of prove that, look, you can do this type of breeding without as much labor as what we do typically at the lab when we're looking at these specific traits. And the Hilo project is another one where we're really focusing on that also, because in the long term, the Hilo project has to be a commercially viable project for the beekeepers. So, and then Ariane might want to say something about the genetic markers and that type of stuff.
Yeah, I mean, I might piggyback off of you, Garrett, if you don't mind. But yeah, I mean, the point is that what we do, or what we're trying to do, both with genetic markers as well as with just standard practices and approach in terms of logistics, is to enable bee breeders, right? So something like a commercial beekeeper, right, who is just really maintaining their colonies for, let's say, productivity of honey or for pollination purposes, like that individual might not have the ability or resources really to be able to breed their own bees, right? Like to a level that accommodates them, right? And right now, there's a really high level entry barrier, right, for anybody else to then take up that undone but much needed work, right, of breeding a bee, right? And so like a lot of what we do is, well, how can we lower that barrier, right? How can we make genetic tools more readily available? How can we make the, how can we do the establish, let's say, the associations, so that when the breeder comes in and says, okay, well, look, like I'm interested in a bee that has low varroa, so that's a varroa resistant trait, it's maybe high productivity, and so on and so on. It's like, how can I get into this, right? Then we can work with them and say, okay, well, what we know has worked well is this approach and this approach. And look, we do have these genetic tools that you can maybe incorporate into your overall pipeline. And so we can work with interested breeders, because ultimately they're the ones that will then produce the queens that the commercial breed or the commercial operations will use, right? So it's more of an enabling role, and that's kind of what the role that we've been working at, honestly, so far. And I mean, maybe more than in certain cases. So for example, we do all the genotyping for the Russian honeybees, and that's an intensive amount of work, but there's nobody else really that would do that, right? Like if you tell somebody, well, I want you to start a company to just genotype these bees, right? They're going to tell you, no, we're going to go bankrupt in 3 seconds, right? So we provide that service, right? And it's something that we probably need to improve, but in terms of like, that's the type of thing, right? So we're talking about these genetic markers, right? Well, if we develop these panels, right? Again, who's going to provide that service, right? A company is not going to be built based on that, right? But if we have collaborators that are breeders, and we can say, well, we help you, whoever, whether it's a single person, like in the Hilo project, right, is David Thomas, who's sort of leading the initiative in addition with all these sort of collaborating groups, or whether it's an association, sort of like multiple beekeepers, similar to what the Russian honeybee has. Like, we can work with them and say, okay, here's how we can enable your process, right, and lower that barrier, so that then when you are able to sort of establish and stand on your 2 legs, you can just continue that on, right? And also in informing, right? So I showed that graph with when we gave VSH queens to honey producers. Well, that turned out to be 2 things. It turned out to be the trait, and it turned out to be their operations as well. I mean, this didn't happen post a study, but what could have happened is a dialogue, if they were interested, or if we wanted to talk more with them, is we could have sat down with them and said, look, this is what's happening to the trait. If you want this trait, or really any trait, and you want to still call your bees this, here's some of the steps that you could do as a commercial breeder, which we know work from other experiences, in order to keep that trait better, right? And I mean, sadly that didn't happen because it was just like a general one-off study, but that's a missed opportunity that we could have capitalized on. And we are now learning from that and capitalizing on it with the Hilo collaboration, and still do with the Russian collaboration. So I hope that answers again. So I'll shut up now because I've been talking too much.
We have another question in here. Which stock right now has the most production of royal jelly? Is there any studies on that? I'm not really sure if we do know that. Kate, you maybe know anything about that? Because I know royal jelly is related to vitellogenin, isn't it? So.
Sure. Actually, I don't think we have any really information on domestic stocks like that, but by far and away, the Chinese royal jelly lines produce a crazy amount of royal jelly. It's just insane. So we know it's something that can be selected, but they have a really unique system where that's kind of what they've been selecting for, and it's almost a closed system at that point in some parts of China, because they really saturated everything, and because they're outcompeted by the native bees, it's really just these kept royal jelly lines. So it's a really neat system, and I think it's a really cool question that I'm getting more and more interested in.
Yeah.
But as far as domestic stocks, I don't think there's really any information about that out there.
Yeah, yeah, and that's what's interesting about the breeding programs done here in the United States and across the globe is that if there's any trait of interest, we could usually breed for it. It's slow and it's difficult at times, but there usually is something that we could do with that. So having a high royal jelly production line would be good if you're doing a lot of queen rearing and queen breeding and things like that. So yeah, these are all specialty things that we could breed for and produce. So Danielle asked about we've done this field day event this would have been 21st years in a row that we've done this event in collaboration with the Louisiana beekeepers now. Usually it's a little bit of a morning session like this where we have like some seminars, and then we actually get out in the field and actually work bees with people. And that's one thing that we do miss this year. So hopefully we'll be able to do that next year. That's something that we keep doing here at the lab, but then also the Louisiana Beekeeping Association does other events throughout the year. So this is just a way that we can connect with beekeepers and maintain those contacts virtually in these restricted times, I guess you could say. So this is a new format they're all getting used to. So I apologize for my little technical ineptitude early on with the audio, but I think we got it fixed and we move forward. So we look forward to getting interaction like this. We usually also send a contingent of people to the beekeeping meetings, whether that's American Honey Producers Association or American Beekeeping Federation. So there's also opportunities to interact with us there as well. And that's actually a really important part of what we do is those interactions with beekeepers, because we get information about problems facing beekeepers in the real world. And that's ultimately what our mission is here, is to solve those or address those problems. So giving us ideas for research is very welcome. And actually that's how we came with the Amitraz Resistance Project. That was after just talking to beekeepers, and I keep hearing the same thing over and over again, and then it finally clicks. So yeah, so these are great opportunities for us to interact with the public, to share our information, but also to solicit feedback from the general public as well. So yeah. Oh, man. Yeah, and so that's what's been interesting about this year is that we had to change how we do research. So research has been very heavily restricted, and when we were able to do it, we have to kind of adapt. So this year for the Amitraz resistance research, we were not able to go on the road, so we had to just mail the kits out and do it remotely. So just got to improvise, adapt, and overcome. So this is something that we need to be agile with and patient going forward. But the good thing is we've been able to do a lot of writing at the lab under these under the pandemic restrictions. So that's kind of nice. And so getting more research out and participating in more events like this. So, but it has put a damper on some of the research we have been doing here. We had, like Mike was mentioning, is that we had our new 5-year research program, and we're all set up and ready to go. And in fact, in late February, early March, we were starting to get colonies ready for breeding experiments and large scale projects. And then all of a sudden we weren't able to do that. So we did lose not a full year, but it's been significantly reduced, I would say. But still, we're able to maintain productivity nonetheless with our maximum telework status. And you'd be surprised how a mountain of data builds up when you can't. We don't have the time to sit up and analyze it as it comes in. So this has been actually kind of we've been making the most of our productivity here. So Sammy asked another question here about Chinese beekeepers test cross with Italians with their native bees, and they call it Italian Chinese bees. Could we get those bees here to produce, to be good honey producers and very healthy colonies? So yeah, that's actually something that's kind of a tricky thing because of importation restrictions across international borders. Even just going across the border to Canada, it's a very, very different situation. So global trade in actual live honeybees can be quite difficult. And like Garrett was saying, even with the United States, shipping, you can only ship semen out to Hawaii, correct, Garrett?
Yeah, that's correct. You can only ship semen.
Okay. So even within the United States, there's even restrictions. So getting bees from China to the United States would be a very arduous task. So when the Russian program started, we were very fortunate in that we had a quarantine facility where we were able to isolate any of those imported Russian bees and keep them out of the normal population. So that took a monumental effort. So this is no trivial matter of importing novel honeybee stocks. It's a long, arduous process because of potential releases of unwanted genetic material into our population. So while it would be an interesting opportunity, it's very good that we have those restrictions in place. So
Yeah, if I can talk a little bit on that also, Frank.
oh,
So I mean, it's not only, it's not only undesirable sort of genetic, contributions, but also like, I mean, it's really, and this is why we had that quarantine facility during the Russian sort of breeding program. It's really kind of monitoring what these colonies are and are capable of, right? So like, okay, that's fine that you're combining these two stocks, but like bees in China, generally speaking, are largely either the endemic Serrana, which is a different subspecies altogether. And so then you're talking about like, well, are you making hybrids or potentially also Italian bees actually? So if you're just taking Italian bees in China and mixing them with Italian bees in the States, like that's still an Italian background. So it's one of those where, the restrictions in there and they're put in place precisely because these stocks need to be monitored and assessed, because we don't want a repeat of something like sort of what happened with Africanization, for example, where, and that was actually a governmentally sponsored program in Brazil, where it was very intentional and it was with the goal of making a better tropical bee. And one hurricane turned it into what we have now 50 years later. So I mean, so this is why we need to be careful, why we're very scrutinous about sort of all these sort of importations. And actually, I would say, you might want to take a look at sort of the APHIS is currently working and making an effort to be more consistent and concrete about what they do and they do not. And I say that because I myself encountered things that are a little bit nonsensical. Like, for example, you can't import bees from territories to the US because they're treated in a different federal capacity and things like that. Or at least initially they weren't, and now they realize that it's the same, so it's not. But these sorts of things are sort of like, both politically charged and sort of practically instituted for reasons. And so it's one of those where, like, APHIS would be the regulatory agents to kind of watch for what is it that they allow and they don't allow. And then we would be the type of agents to talk to say, it's like, well, would this be worthwhile, right? Like, or am I just getting another Italian bee rebranded, which is like paying more money for another Italian bee? Is why? Like, so.
Well, and the good thing is that we're starting to find out that about the genetic diversity of the bees that we do have currently, and what it really comes down to is just building a good stringent breeding program to select from that variation for these traits. So maybe here in the United States, someone wants to start a program to raise bees that produce a lot of royal jelly. That's a great breeding program if you want to get that started. So yeah, there's all kind of options, is just long as you have the genetics of the genetic variability and a strong selective breeding program. We could do that with what we have here, probably out of our domestic stock. Danielle mentioned about talking, if you want to see a video of Bob Danka talking about the VSH program phases, you could go to helobees.com/videos.html and check that website out. Garrett mentioned that it just went live, so that's a good resource about how these Varroa sensitive hygienic bees, this Pauline bees out on the islands, is going to be moving forward.
Yeah, on that, Frank, that website has a lot of information, and they're talking about the phases. There's a lot of history involved, which is not just the VSH, with it being called SMR, then VSH, and then now Pauline, but there was many phases also in the Helo program before it even started, where we were doing research there, which basically, for the sake of time in the talks, I had to skip over a lot of that history that is really interesting, but it's all there on the website. There is tons of information so people can learn more about it. and videos and pictures and data and charts and all that stuff. So it's a really good resource, and it's great to see that it actually is live now.
Good. Yeah, strongly encourage everyone to go check that out to see more information about Hilo bees and Varroa sensitive hygienic bees. Okay, well, it's now noon, and I just want to thank everybody for participating in this event, whether you're a speaker or an observer. This has been great. I wish we could have did it last week, but no one likes a hurricane. So we plan on doing some more events like this. So spread the word, and hopefully we could get more interactions. And very much appreciate everyone's time today. So go outside and enjoy the rest of your day. Thank you, everybody.